Bismillahirrahmanirrahim. Porphyrias are disorders of heme synthesis characterized by accumulation of heme precursors in blood. The word porphyria is derived from Greek and it means purple. It refers to the red-blue color caused by pigment-like porphyrins in urine of some patients with defects in heme synthesis. So, these are rare inherited or sometimes acquired defects in heme synthesis which result in accumulation and increased excretion of porphyrins or their precursors. Inherited porphyrias may be autosomal recessive or autosomal dominant. Each porphyria results in accumulation of a unique set of intermediates according to the deficiency of the enzyme in the pathway of heme synthesis. These porphyrias are classified as erythropoietic or hepatic, depending upon whether the enzyme deficiency occurs in the erythropoietic cells of bone marrow or in the liver. Hepatic porphyrias are further classified as chronic or cutaneous and acute. Acute hepatic porphyrias include ALA dehydratase deficiency porphyria, acute intermittent porphyria, hereditary coproporphyria, and variegate porphyria, whereas the chronic one is the Porphyria cutanea tarda. In contrast to hepatic porphyrias, the erythropoietic porphyrias present in early childhood with photosensitivity. In general, the individuals with an enzyme defect before the synthesis of the tetrapyrrole ring manifest abdominal and neuropsychiatric symptoms, whereas those with enzyme defects causing the accumulation of the tetrapyrrole intermediates show photosensitivity. And this photosensitivity is to the visible light of about 400 nanometers wavelength, that is, their skin itches and burns. There is pruritus when exposed to sunlight due to oxidation of the colorless porphyrinogens to the colored porphyrins, which are thought to participate in the formation of superoxide radicals from oxygen. The resulting oxygen radicals injure lysosomes and other subcellular organelles, releasing degradative enzymes that cause variable degrees of skin damage, including scarring. So now let's review the pathway of heme synthesis and pinpoint the defect at each point. So first of all, glycine and succinyl-CoA condense to form delta-aminolevulinic acid or ALA, followed by the cytosolic condensation of 2-ALA to form porphobilinogen by ALA dehydratase or PBG synthase, which if defective leads to a rare autosomal recessive porphyria. Next, the condensation of 4 porphobilinogen molecules produces linear tetrapyrrole hydroxymethylbilane. The deficiency of the hydroxymethylbilane synthase, which is also called porphobilinogen deaminase, leads to acute intermittent porphyria with acute attacks of gastrointestinal, neuropsychiatric, and motor symptoms that may be accompanied by photosensitivity. The symptoms of acute hepatic porphyrias are often precipitated by use of drugs such as barbiturates or ethanol, which induce the synthesis of heme-containing CYP microsomal drug oxidation system. This further decreases the amount of available heme, and in this case, the photosensitivity may be present though the tetrapyrrole ring has not yet been synthesized. Now, this is due to the spontaneous conversion of hydroxymethylbilane to uroporphyrin 1 and coproporphyrin 1. So, next, the linear tetrapyrrole hydroxymethylbilane is cyclized and isomerized by uroporphyrinogen 3 synthase to produce the asymmetric uroporphyrinogen 3. And its deficiency results in congenital erythropoietic porphyria. This porphyria and all the downstream porphyrias present with photosensitivity due to the tetrapyrrole ring which has been synthesized in this step. Uroporphyrinogen 3 undergoes decarboxylation of its acetate groups by uroporphyrinogen 3 decarboxylase, generating coproporphyrinogen 3 in cytosol. Block at this level leads to Porphyria cutaneous tarda, which is a chronic disease of the liver. It is associated with a severe deficiency of uroporphyrinogen 3 decarboxylase, so liver will be unable to synthesize heme proteins, which it needs to metabolize xenobiotics. Clinical expression of this enzyme deficiency is influenced by any insult or load on the liver, such as hepatic iron overload, alcohol ingestion, estrogen therapy. Estrogen therapy requires heme protein containing mixed function oxidases for their metabolism. Not only that, it may also be precipitated by hepatitis B, C, or HIV infections. The list does not end here. It may also be precipitated by exposure to mere sunlight, due to which the nascent accumulated porphyrinogens may be transformed into colored active porphyrins, which facilitate superoxide radical formation. Mutations to uroporphyrinogen decarboxylase are found in 20% individuals, and the inheritance is autosomal dominant. Clinical onset is in 4th or 5th decade of life. Porphyrin accumulation leads to cutaneous symptoms such as pruritus, itching, as well as the urine that is red to brown in natural light and pink to red in fluorescent light. Coproporphyrinogen 3 enters the mitochondria and two propionate side chains are decarboxylated by coproporphyrinogen 3 oxidase to vinyl groups, generating protoporphyrinogen 9, which is further oxidized to protoporphyrin 9. Block at this level leads to acute hepatic hereditary coproporphyria with photosensitivity. Then block at the level of porphyrinogen oxidase leads to variegate porphyria. And finally, defect at ferrochelatase level leads to protoporphyria with photosensitivity. It may have autosomal dominant or autosomal recessive inheritance. To summarize porphyrias, first we need to summarize the pathway of heme synthesis, where succinyl-CoA and glycine combine to form delta aminoalevulinic acid or ALA. 2 ALA condense to generate porphobilinogen. 4 molecules of porphobilinogen combine to form hydroxymethylbilane, which forms uroporphyrinogen 3, which after removal of 6 carbon dioxide forms protoporphyrinogen, which is oxidized to protoporphyrin, which is converted to heme after incorporation of iron. So here the enzymatic blocks along with their disorders are shown. If ALA dehydratase is absent, there is ALA dehydratase porphyria. If hydroxymethylbilane synthase or porphobilinogen deaminase is missing, there is acute intermittent porphyria. With deficiency of uroporphyrinogen 3 synthase, there is congenital erythropoietic porphyria. Uroporphyrinogen 3 decarboxylase defect leads to Porphyria cutaneous tarda, and coproporphyrinogen oxidase deficiency results in hereditary coproporphyria. Defective protoporphyrinogen oxidase results in variegate porphyria, and finally ferrochelatase deficiency leads to erythropoietic porphyria. Out of these, the common porphyrias are the Porphyria cutaneous tarda and the acute intermittent porphyria.